All right, so we left off last class on this slide looking at a representation of what it looks like where the T cell receptor is having a peptide presented to it on MHC class 2. And because this is MHC class 2 presenting, you have it's a CD4 T cell that stabilizes MHC class 2. And so just to reiterate the point, CD4 is stabilizing MHC class 2. And as we discussed last class, CD8 on the cytotoxic T cells or the CD8 positive T cells, that stabilizes MHC class 1. Yes.
That question 8, which is like I was a little confused with the CD4 stabilizes the contact between CD4 and class 2, but not MHC class 1. I thought that meant that the culture T cells didn't express MHC.
Everything expresses MHC class 1. Not everything expresses MHC class 2. So again, MHC class 1 is on every single nucleated cell in your body. And no one has your MHC class 1. So like it what designates your cells as you. And so every nucleated cell has MHC class 1. Only antigen presenting cells have MHC class 2, because that's what participates in antigen presentation. Does that make sense?
Yeah, I get that part, but I was just confused why it must be stabilizing contact for the MHC class 1.
Because CD4 does not interact with MHC class 1. That's what CD8 does. We have CD4 and CD8. CD4 stabilizes MHC class 2. CD8 stabilizes MHC class 1. And to take a step back and think about the narrative, if every nucleated cell has MHC class 1, if that's like your ID, basically your cell's ID saying it's you, they're also presenting things on MHC class 1 to try, that's from the inside, saying like, am I just constantly assessing cells. This is happening in all of our bodies at this moment. Our cells are presenting things on MHC class 1, and the CD8 positive T cells, those are cytotoxic. If something's wrong with that cell and it's presenting some sort of foreign pathogen there, or some sort of messed up protein, for example, for lack of a better phrase, if someone in this room had coronavirus right now, I know that feels terrible to say out loud, I shouldn't say that. But in their body, their respiratory epithelium has spike protein inside them, because the virus goes inside the cell, hijacks our DNA machinery and using it to replicate and make more spike protein. So there's spike protein inside that's being presented on MHC class 1. CD8 positive T cells or cytotoxic T cells, they're assessing things. And if they see spike protein on MHC class 1, if they're a T cell that's made to recognize that, they'll say, oh, that's not right. I'm a cytotoxic T cell. I'm going to destroy this cell. And so CD8 stabilizes MHC class 1 because those are the cells that are assessing cells on basically on their ID, on their MHC class 1. And CD4 is stabilizing MHC class 2 as that antigen is presented to the T cell receptor. So CD4 is stabilizing MHC class 2, CD8 stabilizes MHC class 1. All cells, all nucleated cells have MHC class 1. Only antigen presenting cells have MHC class 2. Did that help? Okay. So as I just said it a moment ago, we are either our cells, all of our cells are expressing MHC class 1, and only antigen presenting cells are presenting things on MHC class 2. And as you can imagine, the antigen presenting cells are engulfing things that are coming from the outside. Things are exogenous pathogens. Exogenous means generated from outside. Endogenous means generated from inside. So we're going to talk about endogenous and exogenous pathways. So the exogenous pathway, this is where the pathogen is coming from the outside that has been engulfed and is now being presented on MHC class 2. So this pathogen is being digested and broken down within lysosomes. And what's happening here is that it's being endocytosed in the phagosome. And the phagosome and the lysosome, this is a level of detail I'm not asking you all to remember, but I really enjoy talking about it because I think it's cool. That's probably more of an indictment on me than anything else. But the phagosome and the lysosome combine to make the phagolysosome. And what's really interesting here is that it's being digested. It's leveraging the enzymes that are in the lysosome to break down that antigen into smaller peptides. What's going to happen next, and this is important to know, is that there's a separate vesicle where MHC class II is inside that vesicle. So now the phagolysosome is going to fuse with that vesicle where MHC class II is. And there's going to be, it's going to undergo what's called peptide loading, where there's a bunch of different peptides that are available. And those peptides will be loaded onto different MHC class II proteins, and it'll be shuttled out to the membrane to mediate display of that peptide, of that antigen. So we have the entire large antigen that was endocytosed, brought into the cell by either a dendritic cell, a macrophage, or a B cell. It's going to be digested into various fragments using really, really strong enzymes. And then it's going to be fused within this vesicle where MHC class II is ready and waiting to get access to these other peptides from the antigen. So now what's going to happen is we go from this large antigen to small epitopes that are small enough for MHC class II to present it on. And again, these are going to be 18 to 20 amino acids in length. And it's going to present those epitopes on the surface. And it's going to try and find the T cell that recognizes that specific epitope. All right. And because this is being presented on MHC class II, it's going to find the T cell that will match that epitope is going to be a helper T cell that's CD4 positive. And CD4 is stabilizing MHC class II. Now, when the right T cell, the T cell that has been made to its entire fate in life is to find its epitope somewhere. When it finds that epitope that's been displayed to it by an antigen presenting cell, that's going to elicit activation of that helper T cell. That's going to then, if this is a B cell, it's going to send cytokines to activate this B cell to secrete antibodies. But also you're going to have the T cell that's activated, that's going to release cytokines and also going to proliferate rapidly. We're going to talk about that proliferation a little bit later. Are there any questions on this exogenous pathway? Yes. That has MHC class II within it. Yeah. And again, exogenous pathway, this is from an antigen that is from outside our bodies, not generated from us. So for example, this is how, going back to the coronavirus example, this is how the virus, once it's been identified, or if it's captured on the outside, can be endocytosed and broken down. It's not always going to be this. It's not always going to be this process here, but it would be the spike protein that would be presented on the surface here. All right, for the endogenous pathway, this is typically how virally infected or cancerous cells are distinguished, because this is leveraging MHC class I. So this is where things that are, this is the endogenous. Endogenous means generated from the inside. Things that are inside the cell are going to be loaded onto MHC class I, and what's going to happen is that these proteins are pumped into the ER by an ABC transporter, an ATP mediated transporter, and so this is pumping it against. That's important because it's pumping it against the gradient. You're trying to get it into the ER, and so it's requiring that active transport, and it's going to be subsequently loaded onto MHC class I. And so whether it's cancerous epithelial cells or a virally infected cell where it identifies that there's either spike protein or things that are unique to a cancerous cell, that this is a constant surveillance of what cells are doing all the time. And it's going to be loaded out onto MHC class I, and it's looking for its T cell with the cognate receptor for that antigen, the corresponding T cell receptor for that antigen. If it finds the corresponding T cell, and if it's a CD8 T cell, what's going to happen is it's a cytotoxic T cell. It's going to release cytotoxins called perforins and granzymes that will literally poke holes, it'll perforate the membrane and destroy that cell. And so this is how cancer immunotherapy works. They're trying to strengthen the cytotoxic T cells for that specific antigen, or during a virus infection, what we're trying to do is to try and make a lot of these processes more efficient so that it can find the right infected cell and destroy it to eliminate the virus. Are there any questions on the endogenous pathway? Again, endogenous means it's generated from inside the cell. It's pulling things from inside the cell. Yes?
So the T cells that recognize the different peptide on MHC-I, they also have to be specific. Like, it doesn't have to be enough to know that this is self. It has to be, this is not self, and it's this thing that only I recognize.
Yes, that's exactly right, and that's a great question. That's a point I should try and make. I should have made clear. It both needs to be not self and specific to that CD8 positive T cell. Are there any other questions on the endogenous pathway, or endogenous or exogenous? Yes. Can you repeat? I just want to make sure I understood that properly. Can you say it one more time?
Sure. So when
Yes.
there's no region or signal this is always
Always happening. And so you can think of it as a constant assessing of what's going on inside that cell to make sure it's okay, for lack of a better phrase. And so it could be collagen that's being, or not collagen, it could be actin from the cytoskeleton. It could be any kind of intracellular protein that's normally there that could be presented. And the idea is that hopefully there is no T cell that will say there's something wrong here, that this is foreign. And so this is a process that's constantly happening, and it's a way of surveilling for infected cells or mutated cells. So some important things to help keep in mind for the big picture. So each antigen receptor, we've said it usually recognizes one and only one. It usually recognizes a small number of structurally related epitopes. So to continue using the coronavirus example, we're all familiar with new strains and mutations that happen that elicit these new strains. And if you all remember, there's a constant dialogue and questioning of, if we've had these vaccines, will the antibodies generated from them usually work? And the idea is that usually that the antibodies that are elicited are generated from B cells, and they have that antigen receptor for that antigen. They will recognize a small number of structurally related epitopes. So what that means is, if there's a single amino acid change, sometimes what's going to happen is that antigen receptor will still bind. It might bind more weakly, but it'll still recognize that antigen. So for example, during the, I can't remember if it was Delta or Omicron, I think with Omicron, there were, with the Delta strain, there were some changes on spike protein, but what we were able to find out is that the antibodies elicited from the Moderna, Pfizer, Johnson & Johnson, and AstraZeneca vaccines were still able to bind and neutralize the coronavirus from those strains that had mutations from what the spike protein looked like in the vaccine and how it was generated. Now with Omicron, what ended up happening was there were enough changes, and as all of you are intimately familiar with now, if you change enough amino acids on a protein, it can change its conformation. So the question became, now do our vaccines, that the antibodies from these vaccines will they now appropriately recognize this epitope that's structurally related, but now is different in a different conformation. So typically, antigen receptors are very specific, but can bind very structurally related epitopes, both for our benefit, if there is a pandemic like this, or to our misfortune. And we'll talk about this being true with Guillain-Barré syndrome. But the important note to keep in mind is that typically antigen receptors, very specific, and will bind very, very structurally related epitopes. Regardless of the type, once these lymphocytes, whether it's a T cell or B cell, once they are differentiated, they will produce one and only one antigen receptor forever. So when you are born with an innate immune system that's ready to go and interact with any pathogen with any specific patterns that it faces. Our adaptive immune system, our T cells and B cells, we have a huge library of T cells and B cells, with each of them having totally different B cell receptors and T cell receptors, but they will only be available to us if they've seen that pathogen before. So we also call adaptive immunity acquired immunity, because it only works if you've seen that pathogen. All right. And they each have that one antigen receptor. They're each made for that one antigen to recognize one antigen, and we'll have that receptor forever. Yes.
So the reason like there's different flu shots every year is because the flu virus is different, like sufficiently different every year for the T cells that you have that are already that already recognize that particular antigen. But it's different enough that
Yeah.
they can recognize it.
Exactly. Same can be said with colds. There are 200 different types of colds. Once you've had that cold once, your body is immune against it, because now you have T cells and B cells that will eliminate either with a cell-mediated response from T cells or a response from B cells. But because there are I think it's over 200. Each time you get cold, you're seeing a different rhinovirus. It's not structurally related enough. What immunity you've already acquired to be able to protect against it. And so again, yes, Lee. That's a great question. So this is one thing we talked about at the office hours last night, and it is something we'll hint at today, but will make more sense, I think, down the road. But the genetic diversity of viruses and bacteria pale in comparison to our genetic diversity of what antibodies we can generate, or what T cell receptors we can generate. So the number of possibilities, I can't remember the exact numbers, but I'm just gonna I'll just talk about it in more qualitative terms. Our genetic diversity is far more robust than what bacteria and viruses can do. And so we have T cell receptors and B cell receptors that we're born with that are ready to face anything. The issue is that if you're immunocompromised, a lot of times there's an inability to fight against those pathogens. But typically in healthy individuals, you are equipped to respond to any pathogen that you see. And so we'll talk in a few slides, we'll talk about how sometimes that diversity will include autoreactive or self-reactive T cells and B cells, and how we get rid of those. But we are born with a greater capacity to respond to pathogens than what viruses and bacteria can usually make. Yes. Yeah, so you could envision it. The best way to describe this is that over time, we've been naturally selected over time to possess these genes that allow for a greater capacity to respond to different pathogens. And so that's, there's lower order organisms have adaptive immune systems like mice do. And so, when immunologists are studying adaptive immunity, like they'll use mice to study that. But we have a greater repertoire of what can be made to respond to pathogens than what viruses and bacteria can generate. Are there any other questions? These are great questions, by the way. Yes.
So if you have like one specific strain of the cold as like little kid, and then again when you're like 90, how does your body have? Does it just always have like in store like those cells to respond to that?
Yes, and I think that is actually the next slide. So hold on to that thought. The answer is yes, and we'll see how. Yes.
I understand getting the vaccine or like getting the vaccine again makes sense. But if the virus that's out there is mutating, right? Why do we get the same like?
That's a great question. One thing that we have not talked about is that in subsequent exposure, actually we do talk about that, forgive me. In subsequent exposures, the B cells that are already around from the first exposure, this goes back to the question of memory of the cells sticking around after seeing something. You have an ability to create a higher antibodies that have a greater affinity for whatever that pathogen is. So in this case, of a virus. And so when you get booster shots for the coronavirus vaccine, or in response to recognize coronavirus, the booster shots are improving the affinity of your antibodies. And what that's going to do is it's going to help bind and recognize spike protein faster. And with that increased affinity, going back to the first point, because the affinity is so high, it'll do a better job of identifying structurally related spike proteins that might have some mutations. And so the idea is that the booster elicits a response from your immune system that helps make better antibodies that can bind to things better and potentially bind to other things more readily than the antibodies elicited from your first exposure. And so they undergo this process called affinity maturation and class switching. They'll switch classes of antibodies. And after subsequent exposures, the affinity maturation, there will be changes in the genes responsible for making that antibody, so in an effort to create and elicit better antibodies. And so if we, there's, people will joke about umpteenth number of boosters, but the idea is that with each subsequent exposure, you are getting a stronger immune response. And so, for example, before we all had masks, there were probably a lot of pathogens floating in the air that a lot of us have already seen, and your immune system is seeing that and is now eliciting a stronger reaction, or not stronger reaction, that's the wrong way to say it, a more efficient response, so that you don't even feel the fact that you've been exposed to this pathogen, because your body has seen it, it knows it, and it's raising a more efficient response to it, so that you never even feel the symptoms of it. These are all great questions. For the sake of time, I'm going to keep moving, but if questions emerge, please raise your hand. So the ability for only one specific antigen receptor to be present on a lymphocyte explains the specificity of the adaptive immune system and how it's way more specific than the innate immune system. One lymphocyte for one epitope, all right? And so each lymphocyte, its destiny in life is to find its epitope that it was made to recognize on its T cell receptor or B cell receptor. This is going to sound kind of morbid, but in some cases, they will never fulfill their destiny to find that epitope. And so at the end of your life, you may have T cells and B cells that never saw their epitope and are still in a low population, either in your thymus or lymph nodes. And what that also means is that each of us have T cell receptors, or T cells and B cells, that have seen different pathogens and have acquired immunity to different things. But what's also important is that we need lots of lymphocytes to produce the huge wide variety of possible antigen receptors. And this touches back on the diversity that's present in our immune response. So we are going to talk quickly about the origins and locations of lymphocytes. and their precursors. The word precursors typically is referring to the cells that came before them in their lineage or differentiation process. So, stem cells are an example of a precursor to a lot of cells. And there are lots of different kinds of stem cells. So for example, we think of embryonic stem cells become maybe hematopoietic stem cells, which can become a red blood cell or a macrophage or a T cell or B cell. So the precursor for all of those cells are embryonic stem cells. And so it's kind of like one step back up in the differentiation process. And we'll talk about the differentiation that's independent of antigens, that happens before you ever see an antigen, and the differentiation that happens in response to an antigen, dependent differentiation. So all of our immune cells are derived from the HSC, the hematopoietic stem cell that is in our bone marrow. There are a lot of people who have leukemia that you might hear that they get a bone marrow transplant because leukemia is a cancer of our specific immune cells. And oftentimes they need a new population of immune cells to get rid of that cancer. And so they're getting a new source of hematopoietic stem cells. And so hematopoietic stem cells are found in the bone marrow. And they can become the common lymphoid progenitor. I'm not asking you to recognize every single cell in this process called hematopoiesis. You probably remember from the pre-lecture, that crazy tree of cells. It's an effort to get you to appreciate what the process looks like. And so in the bone marrow, the common lymphoid progenitor can become a B cell. For T cells, the common lymphoid progenitor will go to the thymus and mature into a T cell there. So to help you memorize this, B for B cell matures in the bone marrow. T for T cell matures in the thymus. So they will both originate in the bone marrow because that's where hematopoietic stem cells are from, which becomes the common lymphoid progenitor. And from there will be specific cues for the common lymphoid progenitor, the CLP, to leave and go to the thymus to become a T cell or to become a B cell in the bone marrow. And from there, so these are our primary lymphoid organs or tissues. I promise I didn't do that. There we go. We'll wait a second for it to come on up top. There we go. So the bone marrow and the thymus are primary lymphoid tissues because those are the two tissues where B cells and T cells will differentiate into. And so from there, we have secondary lymphoid tissues where B cells and T cells, once they've matured, will go to. So the lymph node is a really common secondary lymphoid structure because those are the sites typically of immune cell education where they will see antigens or see antigen presenting cells presenting different epitopes. And so other secondary lymphoid tissues are the appendix and also your tonsils. And so as you can tell, some secondary lymphoid organs can be taken out, like the appendix and tonsils. But we have so many different lymph nodes throughout our body that those are oftentimes the most important secondary lymphoid tissues. So for antigen-independent differentiation, this is where differentiation occurs without the need for antigen. We'll have these hematopoietic stem cells that give rise to many, many millions of antigenically committed lymphocytes. What that means is that we're going to have millions, hundreds of millions of lymphocytes that each recognize one and only antigen, and they have either the T cell receptor or the B cell receptor, the antibody, that will recognize one and only one epitope, usually. And so the generation of these are naive. They have not been activated. They are committed to being a B cell or T cell, but they are naive because they have never seen their antigen before. These naive lymphocytes are present at birth in the primary lymphoid tissues, and their presence there does not require antigen. And so when I've talked about this library of T cells and B cells, this is exactly what I'm talking about. That at birth, you have a whole host of T cells and B cells that are all unique in what their antigen receptors recognize, but they're naive because they have never seen their specific epitope before. And so they're poised and ready to go, but they're still naive. And so this is the acquired immunity part, where their capacity to either make antibodies or have a huge reservoir of T cells or B cells ready to go are not available yet because we've never seen these antigens. Whereas macrophages, dendritic cells, the cells of the innate immune system are in our tissues and ready to respond to pathogens should they face them. Are there any questions on the previous slide or antigen-independent differentiation? You guys are asking great questions today. I want to keep it going. Yeah. Yes, that's actually a great question. So some antibodies will cross the placenta. And so in utero, typically there is some semblance of immunity conferred upon the child. And so, for example, like my wife got the coronavirus vaccine when she was 5 months pregnant. And so our son was born with those antibodies. And so there's a lot of studies that have confirmed that that because we have a new vaccine technology, that this actually helped pass on antibodies to the newborns that has been known with other technologies, but now is also demonstrated with the new lipid nanoparticles. And so, or the RNA-based technology. And so typically it's passed along that way. It can also be passed along through breast milk. And so there's the maternal newborn crosstalk in immunology is wild. And there's a lot that's not known. We have a new professor in the department, Sapita Dolatashi. Did I say it right? Dolatashi? Dolashahi. She's gonna kill me. She is using computational modeling and bioinformatics to really study the maternal newborn immune system crosstalk in really compelling new ways. Because if you think about it, our immune system is meant to recognize what is self and not self. A newborn in your belly is not self. That is a totally different person. Yet women typically do not develop an immune response to their fetus. And so there's this really super fascinating that they can pass on their immunity without developing an immune response to their fetus. And so we have a great professor here that's at the forefront of that. So positive and negative selection of T cells. And so as I described, we develop a ton of antigen-specific lymphocytes, but it's possible that some of them are either defective or autoreactive, self-reactive. And so we have ways to select out those T cells so that they don't persist. So positive selection, what we do here is in the thymus, after these T cells have matured to become naive T cells, they're in the thymus now. What ends up happening is that we want to keep only the T cells that have a T cell receptor that adequately binds to MHC. And so what ends up happening is that we have cells in our thymus that kind of test and interrogate which T cells properly bind to MHC, to their respective MHCs. Those that do not will undergo apoptosis, programmed cell death. And so I always really enjoy kind of demonstrating apoptosis, but basically it's kind of someone like at the front gate, Scott Stadium letting people through, and the ones that don't properly interact with MHC class are people trying to sneak something into Scott Stadium. And so they're gonna say, okay, you're done, you can go through, And so these T cells that are red are undergoing apoptosis and dying and not sticking around, whereas the rest do. And so now what we're gonna do is we're gonna induce apoptosis on those cells that bind too well to self cells. So the cells that are autoreactive and binding too strongly that might induce an autoimmune reaction, those cells will now undergo apoptosis and die. And so this is making sure that we have T cells that properly respond to MHC and that do not have a strong interaction with our own cells. And so what ends up happening is because we're able to make so many, what ends up happening is that about less than 5% of these T cells are the ones that make it through positive and negative selection. And so we maintain our diversity at the expense of some self-reactivity. And this same process is what B cells undergo. Yes.
So when cells are being tested to see if they'll bind, is the testing tested that they consistently bind when to class 1 and 2, or are they also, are these testing the cells also when
Not exhibiting antigen, not presenting antigen. This is interrogating a proper interaction between their T cell receptor and their corresponding MHC. That's a great question. Are there any other questions? All right. So for clonal selection in this diversity, so once these lymphocytes have seen their specific antigen, in the case of a T cell, we'll focus there. Once a T cell has had the epitope that it is meant to recognize, its cognate antigen, its corresponding antigen to its T cell receptor, once they become activated, they will proliferate rapidly. So much so that when this is happening in your lymph nodes, typically when you have an infection, you feel your enlarged lymph nodes. This is what's happening. You're having your T cells and B cells proliferate remarkably. And so you'll have a degree of doubling where the single T cell that recognizes that antigen will continue to double and continue to double, where you have approximately 2 to the 20 doublings. And what ends up happening is that then those T cells will be prepared to go to the site, to drain out of your lymph node through the lymphatics and go to the site of infection or where that pathogen is. So this is, now we're going to look at antigen-dependent differentiation. So to this point, the antigen-independent differentiation was the development of naive T cells and B cells that that now are prepared to respond to their specific antigens. Once an antigen binds to that specific lymphocyte that it is meant to be recognized by, we'll produce what are called fully active effector cells. Effector cells are cells that mediate the response. All right, naive cells are just sitting and waiting for their specific epitope. But once they are activated, they'll become an effector cell. And so they're no longer just simply a T cell or a B cell. B cells will become a plasma cell. Those are the professional antibody manufacturers, where in the pre-lecture, there was an electron micrograph image of a B cell next to a plasma cell. And you can see the huge dramatic change in the amount of rough ER they have to crank out as much antibody as they can. T cells are more complicated. And this is a 30,000 foot view of it. And so we talked about how CD8 positive T cells are cytotoxic T cells. They will kill self cells that are presenting some sort of foreign antigen on MHC class 1. So the way, instead of, we call them either CD8 positive T cells or cytotoxic T cells. We also have helper T cells. They will induce other cells, either T cells, B cells, and oftentimes other cell populations as well, to differentiate and proliferate, and they're CD4 positive helper T cells. Yes. So that's a good question. So we typically have multiple, but it's not, it is typically mediated by cues in the lymph node that help maintain a certain number across our body. And so for simplicity's sake, we show one and only one, but it's not just a single cell. And so the last cell below the helper T cell is one that we have not talked about yet, called the regulatory T cell. And so this is a cell that can suppress the immune response to common antigens. So for example, they are the brakes on parts of our immune response. And so our bodies are constantly assessing for foreign antigens, but we also eat things that are possibly that are foreign antigens, are not of self. And so our gut it's debatable, might be the most immune cell rich organ in our body. And Tregs play an important role in providing what we call tolerance, the ability to see a pathogen and not attack it, to things that we're constantly eating. So a lot of times food allergy is partially attributed to regulatory T cells that are dysfunctional and that are not helping to pump the brakes on an immune response to things that aren't worthy of eliciting an immune response to. And what's also worth noting is helper T cells are also true of CD8 positive, but particularly with helper T cells, excuse me, are further differentiated into, I think we're sitting at 5 different types of helper T cells. There's also immunologists that are constantly wrangling to like define a new T cell. But as of now, we have several different kinds of helper T cells. But the take-home message on this antigen-dependent differentiation is that B cells will become plasma cells whose job is to pump out antibodies. And T cells can become cytotoxic T cells, the CD8 positive ones that respond to self cells that are presenting a foreign antigen, or helper T cells that help produce an immune response to help mediate a response to that pathogen, or regulatory T cells that pump the brakes on common antigens that do not merit a robust response to. And so this is not dissimilar with B cells, where their activation is where their membrane antibodies, their B cell receptor, will bind to an antigen. And what will end up happening is, in combination with cytokines from helper T cells, when they send signals to each other during antigen presentation, B cells will become activated. They will differentiate into a plasma cell. And what will end up happening is that some of those plasma cells will go on to proliferate and crank out antibodies. Some of them will be designated to stick around as memory cells, so that when we see a subsequent exposure of this same antigen, those memory cells are there and available to release antibodies to a greater degree than when it was first seen. And this is also true with T cells as well, where in response to a primary exposure, you're going to get robust proliferation. Now, what's important to note is that when they become these effector cells, some of them will go on and do the work of eliminating that pathogen. Some of them will maintain as memory T cells that are prepared to respond to a subsequent exposure to this same antigen. And so should that happen, if there was a second exposure, what will end up happening is, in the way we've described it here, instead of starting with just one T cell here, now you have a multiplied response, because now you're starting with more than that one single T cell, and you're able to elicit more T cells rapidly, as well as a larger pool of memory T cells that are going to be even more efficient at responding to this pathogen in a subsequent exposure after this. And so these memory cells, the key thing to note here is that they are providing an enhanced response to a previously seen antigen here. So all of this circling back to autoimmune disease. And so autoimmune diseases, while each single one has a somewhat not significant prevalence, altogether, what you begin to see is that autoimmune disease altogether is a really big problem. And so celiac disease is an autoimmune attack on the intestinal wall, and it's exacerbated by the intake of glutens. Type 1 diabetes is an autoimmune attack on our own pancreatic beta cells. And so those are the insulin producing cells. And so type 2 is usually not responding to insulin, whereas type 1 diabetes is that you do not even have insulin because the cells that make it are seen as the antigen and destroyed. Multiple sclerosis is where the demyelination occurs in a response to an autoimmune response and is targeting our central nervous system, specifically the brain and the spinal cord. And finally, rheumatoid arthritis, where typically the connective tissue proteins in our joints are typically the foreign antigen and attacks our joints. And so over time, what you begin to see is while each one specifically might be up to 1% of the population, if we start to list out each of these autoimmune diseases, they'll all add up to a really significant portion of our population. And so being able to understand and address these are really important. So a possible etiology for autoimmune diseases is what we call molecular mimicry. What that means is that when we have an antigen that is initiating an immune response, what happens is that there's a cross-reactivity of that specific antigen where it mimics one of our own self proteins. And so this is one school of thought where sometimes the foreign antigen resembles something that's within us and will then elicit an autoimmune response on what it mimics within our own body. And so now at the end of this, we have antibodies or T cells that are now identifying some innocent self-antigen as now an antigenic immune-inducing pathogen. And so typically what can happen is that the protein might be inhibited, or in other cases, like with type 1 diabetes, the expressing cells of that protein can be targeted and destroyed. So taking all of this back in the last few minutes to Guillain-Barré syndrome, we described how most cases begin a couple weeks after an infection. And that was usually in response to C. jejuni infection, bless you. It causes what we call enteritis, that is inflammation of the small intestine. And so it's been demonstrated pretty clearly that mimicry is the established cause. And so what's important to know is that we have these proteins. The autoantigen here is what's called, sorry, not the autoantigen. What's important in the pathogenesis is a protein called CD1. So we talked about how MHC class II presents peptides during antigen presentation. I'm teaching you all the foundational components of the immune system. What's also frustrating is that the immune system is not very clean and does not always obey its own rules. So CD1 is a protein on cells that will actually present lipids. So MHC class II typically presents peptides. CD1 will present lipids in an antigen presenting way to CD1 responding T cells. And so what will end up happening is that when C. jejuni has been ingested and we have an infection, what will end up happening is that it'll be recognized by B cells, it'll digest it, present it, but what ends up happening for the peptides, it'll be presented, but for the lipids that make up the C. jejuni bacterium, there are these sugar-rich lipids called lipooligosaccharides that are presented on CD1 to T cells. And so these CD1 reactive T cells that are made to recognize that lipid will be activated, and now the B cell and the T cell will both be activated, and now we have antibodies against that are recognizing lipooligosaccharides. What's important to know is that those lipooligosaccharide antibodies are now going to be identifying a component of our own cells called gangliosides. This is the autoantigen in Guillain-Barré syndrome. They're what's recognized as foreign because of their structural mimicry to C. jejuni. So this is the lipooligosaccharide that the antibody is usually raised against, and this is the ganglioside that is on our Schwann cells in our peripheral nervous system. And so it's enriched at sites where there's a lot of signaling, and so the antibodies will bind to the gangliosides and produce an autoimmune response. And so what ends up happening as a response to this is that antibodies are recognizing the gangliosides on our Schwann cells, promoting demyelination of our nerves, and producing all of the downstream neuromuscular effects that are presented in Guillain-Barré syndrome. So treatments, we talked about how there's an issue with breathing, and so there's typically respiratory support that these patients are put on. They're usually put on some sort of what mimics the iron lungs of old, but now what we typically use is a ventilator And oftentimes this can be problematic because, as has been reported a lot of times with patients that have coronavirus, there's also a lot of ventilator-induced lung injury. They'll also be put on immunosuppression therapy to try and dampen that immune response. And they'll also use what's called plasmapheresis, as we remember from electrophoresis, where we're moving, the pheresis implies a moving. What we're doing is we're moving their plasma to get rid of their antibodies to try and purify those out, the ones that respond to gangliosides. In addition to that, we can also use immunoglobulin antibody injections to try and neutralize those autoreactive antibodies. We have a great group of people that study adaptive immunology here, as well as some really big names in the field that have made really important discoveries. And I encourage you to either look at the people that I've listed here or to look at the Carter Immunology Center, which is that way in MR6. And we have a great group of people. With that, that concludes our lecture. I'm sorry for running a few minutes over. I'll be sticking around if you have any questions.